Hello, IB Bio students. All right, so today we're going to continue 8.3 and we are going to look at photophosphorylation. So here's the essential understanding. Reduced NADP and ATP are produced in the light-dependent reactions. So we saw how this happened in the light-dependent reactions. So these molecules here, NADP and ATP, are the molecules that are able to carry the chemical energy that was converted from light energy from the sun, right? NADP gets reduced to NADPH. Um, to NADPH. And ADP gets photophosphorylated. So then these two molecules here are going to be used in the light independent reaction later to produce glucose, which is what the plant is trying to do first and foremost. So the production of ATP by the light-dependent reactions is called photophosphorylation because it uses light as an energy source. Now today we're going to talk about how this photophosphorylation may either be cyclic or a non-cyclic process. So we're going to look at the difference between the two. So let's move down here and let's look at cyclic photophosphorylation. So again, let's pay attention to what we are seeing here. So here we have the thylakoid membrane. Here we have the stroma, which is the cytoplasm of the chloroplast, and here we have the inner or the interior of the thylakoid. All right, so in cyclic photophosphorylation, this involves only one photosystem. So here you're looking at photosystem 1. And it does not involve the reduction of NADP+. So remember let me move here to this other slide. Remember when we saw the details of the light-dependent reactions, and we saw this excited electron here get excited, and it would help reduce NADP+ to NADPH. Okay, remember that? Okay, this is not going to happen here in cyclic phosphorylation. So this electron does not get transferred to NADP+ to get reduced into NADPH. So instead, this electron goes back to its own electron transfer chain. So this right here is the electron transfer chain. So what is happening when this electron moves along this electron transfer chain? Okay, between the photosystem and this transmembrane protein. So when the electron moves down this electron transfer chain, it is going to release some of its energy because, again, it's going to move down the electron transfer chain. It's going to release some of its energy, and some of that energy is going to be used to pump these hydrogens from areas of low concentration to areas of high concentration. Okay, so here's the hydrogen that gets pumped in using the energy from the electron transfer chain. Now, what's going to happen to these hydrogen ions? They are going to move, again, via diffusion through ATP synthase. So here we see ATP synthase. And that's what's going to generate ATP, okay, through chemiosmosis here. So notice that in this cyclic photophosphorylation, right, the photosystem 1 restores its own electron supply. So it's not getting electrons from photosystem 2. It's using its own supply of electrons. So the de-energized electrons that come back to photosystem 1, they go to a higher energy state by more light hitting this photosystem 1. So we call that cyclic because the electrons are returning to the same photosystem. So the photosystem 1, it's restoring its own electron supply. So it goes in a circle like that. It goes in a cycle like that. So we call it cyclic photophosphorylation. ATP being produced here is through a cyclic flow of electrons along photosystem 1. Okay? NADP plus is not being reduced and water is not needed. If we look back when we studied the light dependent reaction here, we saw that water was needed right here to replenish the electrons in photosystem 2, right? And we saw how these electrons aided NADP plus to get reduced into NADPH. But we're not seeing that in cyclic photophosphorylation, right? So then what happens in cyclic photophosphorylation? Well, this is what we learned initially. Non-cyclic photophosphorylation, sorry, I said cyclic before, but I meant non-cyclic photophosphorylation, right? This is what we learned before. So in this type of photophosphorylation, we use the two photosystems. We use photosystem 2 and we use photosystem 1. Notice how NADP plus does get reduced into the carrier molecule NADPH, right? So this electron in photosystem 1 that got excited does get transferred here, and it reduces NADP plus. And here we see that when light is absorbed into photosystem 2, the excited electrons will go down this initial transport chain, right, between photosystem 2 and photosystem 1. And that's what's going to replenish the electrons in photosystem 1. And here we see the photolysis of water releasing electrons and replacing the electrons in photosystem 2, right? And then you get your oxygen byproduct. So non-cyclic photophosphorylation involves the two photosystems, whereas cyclic photophosphorylation only involves the cycling of these electrons down its own electron transport chain, right? Electrons are not donated to NADPH. So here let's look at a comparison of the two. So in cyclic photophosphorylation, only photosystem 1 is involved. In non-cyclic, both photosystem 1 and photosystem 2 are involved. In cyclic, water is not required. In non-cyclic, water is required. In cyclic, oxygen is not evolved or produced. In non-cyclic, oxygen is evolved or produced. In cyclic, NADPH is not synthesized or reduced. In non-cyclic, NADPH is synthesized. And in cyclic, it is used to produce additional ATP in order to meet the cell energy demands. And in non-cyclic, products can be used for the light-independent reactions. So there is enough ATP here to make the products in the light-independent reactions, which is glucose. So let's talk a little bit about this additional ATP requirement. Usually in plants known as C4 plants, and these are plants that are found in tropical, elevated regions where there's really high daytime temperatures and droughts. So these C4 plants only make up about 3% of plants worldwide, but they have an additional ATP requirement. Usually they require 3 ATP molecules to 2 NADPH molecules. So there's a higher ATP requirement in these types of plants. So these types of plants, they're going to have to, besides undergoing non-cyclic photophosphorylation, they're going to have to also undergo cyclic photophosphorylation to produce an additional amount of ATP that the plant actually requires. And notice that since these plants are usually found in drought areas, cyclic photophosphorylation, since it doesn't require water, then that is a good thing, right, for this cyclic mechanism to happen. So here we see a comparison of cyclic and non-cyclic photophosphorylation. Notice how in cyclic only photosystem 1 is used. In non-cyclic, both photosystems are used. Notice how in cyclic, the electrons that get excited are then used in the electron transfer chain to pump hydrogen in and then out to form ATP, right? Whereas in non-cyclic, those electrons are donated to NADP+ to form NADPH, right? So both mechanisms are a little different. So here is an overview of the light-dependent reactions, both cyclic and non-cyclic. I'm going to play this video for you. Uh-oh, it looks like it doesn't want to play. Okay, it looks like it's really slow, so you guys can play it on your own in BioNinja. The other thing that I wanted to go over is in Khan Academy, they also have an explanation of cyclic and non-cyclic photophosphorylation that is very good. Okay, here they show you photosystem 1 and photosystem 2, and they go into a little bit more detail of how these photosystems work. So if you want to look at it, it's a very good resource. And then they go into a little bit more detail as to why the cyclic pathway exists and it not just the non-cyclic pathway in plants, right? So we know that sometimes, look at what it says here, the ratio of NADPH to NADP+ is too high. So then some plants need to focus on cyclic photophosphorylation only to produce ATP. Also, like I was saying before, some photosynthetic cell types require really high amounts of ATP to synthesize the sugars in the light-dependent reactions, and we call these the C4 photosynthetic plants. And finally, cyclic electron flow may play a photoprotective role, preventing excess light from damaging photosystem proteins and promoting repair of light-induced damage. So if you, again, want to get a really high amount of ATP production, you are going to undergo a cyclic electron flow or a cyclic photophosphorylation along photosystem 1, right? So you're not going to use photosystem 2, and you are not going to reduce NADP, right? So this is just going to help create that proton motive force inside the thylakoid, right? So that all of these hydrogens can accumulate inside the lumen of the thylakoid, and all of these hydrogens, when they diffuse back through chemiosmosis, can generate a lot of ATP. All right, guys, so that's it for today. Make sure you review this material, and I hope you have a great day.